Day, Homo sapiens. Welcome back to the history of microbiology. I mean, the book written by the Prescott, and we are finally at the stage where we are not stuck at the landmarks in the history, and we are just going to study the discovery of microorganisms and the major scientists that were involved in that. So, wait, let me check if my live is working because I don't want it to trouble, and I will be doing this, you guys can see it with me, right? Wait, let me refresh it, and I just want to know if it will work. Give me a second. Okay, so it's working just fine. So let's turn this off and let's go back. So we were talking about studying the discovery of microorganisms, right? And if you are curious what we studied in the landmarks, in the history landmarks of history, we were actually studying the big discoveries, including the scientists, which were not that major as compared to few of them, and comparing them with the world history, right? And it wasn't just the big history, it was the major history occurring in the world in the past. So today we are going to almost summarize it in this paragraph, the discovery of microorganisms. You can watch the history in my other lives in the same playlist. And so let's start, let's begin. Are you guys ready? And okay, so I think people watching this video have no idea what I am doing, so it's just me reading the book and explaining the terms I know. And I am doing this because I know that the students or anyone, like it's less habit, it became less of the habit to read the books and study from them. Instead we just go to the Google and just took the material from that. So I want to read this book, and if anyone other than me is interested in reading, so good luck to you as well. We are going to cover this book, I don't know, maybe in a year, maybe in 2 years, or maybe in months. Who knows if I stream more than 1 hour, or and by the end of it I am sure that we will be masters in the basics of microbiology. And it's not just for students, okay, it's also for people just want to read book and the history of microorganisms. So anyway, without further ado and wasting time, let's begin. I have been blabbered a lot, so, and also don't expect me to know everything. It's more like I know nothing, and that's why I am reading it with you guys, because, well, we all may be wanting to read this book because we want to get the knowledge. So the discovery of microorganisms. Okay, before going to that, sorry, I'm cutting my own concept again and again. I just wanted to ask if the window will work, like, is it easy to read in this way, or the previous one was okay? I thought reading like this would be okay because it like the handwriting is bigger, and I thought it will be easy, but then again it just becomes few lines, and I think that would irritate some people because we are used to seeing the bigger pages instead of just few lines. So definitely tell me which one is better. Even if you don't watch the video, just tell me the suggestion if this one works or the full page picture one works. And thank you for telling me in advance. Even before microorganisms were seen, some investigators suspected their existence and responsibility for disease. Okay guys, there's one thing that's missing in this book, and that is the talk of miasma, right? Miasma or bad air. Even before these 1400s or 1500, it's the talking about who knows, I think it's before even 10th 100, no sorry, thousands. So it was the miasma or bad air. People were getting disease or people were getting infections because there was something bad in the air, and that was actually the precursors that there were microorganisms, but they didn't know. They just thought that the air was bad and dirty, and that's why it was causing diseases. So I think this one just skips that and come further, forward. So I just wanted to add that. Among others, okay, so they are saying that before the microorganisms were seen, we already were able to, sense them in a way. So among others, the Roman philosopher, look, what? Lucretius? Lucretius, guys, I'm not good at, uh, you know, pronouncing the names. So if you know the correct pronunciation and I'm pronouncing it wrong, correct me. About 98 to 55 BC, and the physicians Girolamo Francastoro. So these two scientists, Lucretius and Girolamo Francastro, 1948 and to 1553, suggested that I think this is the age, maybe their age, the one that they lived. Is their life age or is the history age? I think it's their life. Suggested that the diseases were caused by invisible living creatures. Okay, so they were this close. They already knew that they were living creatures, but they were just invisible. The earliest, guys, just think about it. Someone telling you, okay, sorry, I think you didn't saw the lines. Guys, imagine someone telling you that, yeah, you are getting diseased by something invisible. I think people at that time would be laughing at you. Like, how the heck would something you can't see cause that? People don't usually accept things that they cannot see, even though they have feelings which we cannot see, and other things like heat radiations and things like that. The earliest microscopic observations appear to have been made between 1625 and 1630 on bees and weevils by the Italian Francesco Stelluti. Okay, sorry. So, okay, the microscopic observation. The microscopic observation means it was using the microscope, and it was this scientist, Francesco, in 16. Okay, see, this already jumped to the 1625, and when we were studying the landmarks of the history, we studied that in something about in 1500 the microscope was, the simple microscope, compound microscope was mad, and I think it's because of that that Francesco Stelti could, see the bees and weasel parts under the microscope in a bigger, frame. In 1665, the first drawing of the microorganisms was published in Robert Hooke's Micrographia. The first drawing. Guys, let's see. I know about micrographia, right? But we don't know about the how was the first microorganism in micrographia. Let's see if they can tell. Where is it? Is it this one? Wait, is it fungi? Is it fungi? No. We don't want this. Okay, so it was the fungus. The first microorganisms depicted in the published work was the microfungus mucor, which appeared in mucor. Are we familiar with the mucor? Where does it grow actually? I'm sure we would know. Okay, soil, plant debris, stored grains. Okay, guys. You have seen it. I'm telling you, you have seen it. These things, these whitish like things, I'm sure you have seen it on your food or trees, or like the things that have time passed on them. Yeah, so it was this, and look at this, how beautiful does it look. even though it would be so dangerous. Where is it? Here. Look at it, so white. Okay, so the first one was this. Okay, we are losing our track, and we were not trying to look at this. We were trying to look at the first microorganism and how it was drawn in the Micrographia. It was the book. Okay, look, this is the book, Micrographia by Robert Hooke. Robert Hooke is actually the one that discovered the cell, even though he couldn't name it the cell. So where is it? I think it is this one, right? Yeah, or maybe no, I think it's this one. Okay, so however, the first person to publish extensive, accurate observation of microorganisms was the amateur microscopist at Antony van Leeuwenhoek. Guys, this is the name. This is the father of microbiology. Remember his name, Antony van Leeuwenhoek. The father of microbiology came, and they said that he was amateur, like he wasn't even expert at that, and he already observed microorganisms. I think he was just curious, like what was happening in the water, what was happening into the soil, and what were the things that were happening. And he, like a child, maybe put the things on the microscope, and then he found out, wow, there are something that's moving, and when I take that out from the microscope, I can't even see that. So these are the invisible. So he published it. He just didn't see it, he published it in 1632 to 17. Okay, so this is just his life, lifespan of the Delft, the Netherlands. Should we search where the Netherlands is? Why not? Don't get bored, okay? This is just the study together, so we can search about things like that. Don't be so fixated on one thing. That's why we don't know about the world's map or the world's knowledge, because we just study one thing. I mean, it's a good thing. What? The Netherlands is such a big country. I didn't know. I thought it would be something smaller. Wait. On map, which continent? Sorry for my lack of knowledge on world maps. I'm sure you guys wouldn't know that as well. Okay. So it's in the Europe here. Look. Okay, so we now know that this guy was from Europe. Leeuwenhoek earned his living as a draper and haberdasher, a dealer in men's clothing and accessory. Okay. But spent much of his spare time constructing a simple microscope composed of the double convex lenses held between two silver plates. Guys, how is a man that's working in a clothing shop and accessories shop has mind to create microscope and putting glasses lens and silver plates and then even using it? Wow. Intelligent people has intelligent hobbies. His microscopes could magnify around 500 to 300 times, and he may have illuminated his liquid specimens by placing them between two pieces of glass and shining light on them. Okay, guys, so the two pieces of glass they are talking about is the slides. These days we use the slides, the rectangular shaped things. Wait, let me show to the people who just don't know. And by the way, no. If you guys are studying microbiology, of course, how can we know? How can you have not seen this? See these transparent things. These are the slide and that we put. And I think it's because of their lack of knowledge, they were calling it the pieces of glass. And he said that he made the specimens liquid, or they were already in a liquid shape. So, and he was placing that between the pieces of glass and shining light on them at 45 degree or at a 45-degree angle to the specimen plane. Okay, I think he just found out that moving the light around, he found out the exact spot where he thought that this angle works, and I think he used that the same angle over and over again. This would have provided a form of dark field illumination. Wait, what is this thing? I totally don't like this. Yeah, provided a form of dark field illumination in which the organisms appeared as bright objects. Okay, dark field illumination. So he kept everything, like I think his slide was black. No, against a dark background. Yeah. These days our slides are transparent because we put colors, or you can say we stain our microorganisms, the invisible creature. We stain them and it appears in a colorful form. But I think this time he didn't know the staining processes. It came very much in the future. So he instead made the background darker so the invisible things could appear just fine, and they appeared as bright objects and made the bacteria clearly visible. Beginning in 1673, we are jumping, but still in 1600s, Leon Higgs sent a detailed letter describing his discovery to the Royal Society of London. Wait, Netherlands? He was from Netherlands. But why did he send his discoveries to the Royal Society of London? Is London in Europe? London. I think I might be the first person that don't know where the London is. Right? Laugh if you want. It's funny, of course. Wait. London, England. I don't get it. Continent. What? It's in Europe as well? United Kingdom. UK? Europe. Okay, yeah, it's the capital. Wait. Is London capital? Is London capital of? UK. Guys, why don't I know it? Capital of England. And is England in UK? Of both England and United Kingdom. Wait. How can it be? Is UK? Is England in UK? United Kingdom. United. Is the island country in the northwest Europe. England is the largest country in the island country. The capital of the England is London. The UK joined European Union, which was originally the Commune. How can a city belonging to the How can a United Kingdom be a city? Okay, it's a kingdom. It's a kingdom. I think they have a lot of countries. Okay, for now I am getting this, that the UK is a bigger one than the England and then the London. It is clear from his descriptions that he saw both bacteria and protozoa. Like now that he described it, it's a bacillus shaped, or round shaped, and for protozoa maybe of course the same definitions, or and these days we can get what this guy was talking about. And as important as Leon Hooke's observations were, the development of microbiology essentially languished for the next 200 years. Guys, so there was a break Like, people either didn't care about the published document, or people just didn't think it was that, important, maybe, or they didn't know the significance of it. So the work was languished for the next 200 years. Little progress was made, primarily because the microscopic observation of microorganisms do not provide sufficient information to understand their biology. That's right, not just their biology, also we can't understand their function through just a microscope. And the microscope that is simple enough that we can just put the light and it just 30 to 500 times isn't that big. So, for the discipline to develop techniques for isolating and culturing the microbes in the laboratory were needed. Yes, we needed the command over the isolation and identification of the microorganism, because it wasn't like we are always going to get a random sample and we are always going to get the random bacteria, right? We needed to know what bacteria we are isolating and what bacteria are we growing. By culturing means we are growing, multiplying them. And of course, for that we needed lab. Many of these techniques began to be developed as the scientists grappled with the conflict over the theory of spontaneous generation. I blabbered about this spontaneous generation in my previous video a lot, and this is a failed theory nowadays, right? It was rejected quite back in the time, and this stated that the life originated from nothing, like it was spontaneous. And we now know that life comes from life. We know that there must be something in the back if we are seeing a living being. That means there must be a parent of that living being from which it, produced. And in spontaneous generation, they couldn't see the microorganisms. Like, they couldn't see the eggs. They were microscopic. And then suddenly the flies came out of the meat. So they just thought that it might be out of nothing. This conflict and the subsequent studies on the role played by the microorganisms in causing disease ultimately led to what is now called the golden age of microbiology. So even if it was a conflicting concept, the spontaneous generation, and people rejecting it over and over again, and so the research was still conducting. It was a good sign that people were, paying attention to something like this, something that is not even a success in the current days, but it was a big thing in the previous time. So so it played a role, and by microorganisms in causing diseases ultimately led to a golden age of microbiology. They are not talking about nowadays, okay? They means that after studying different microorganisms in diseases, etc., now after this spontaneous generation, or whatever this age was before this, that was over. Now the microbiology was starting to dominate the world, and that was the golden age of microbiology. And from this golden age, we are getting our scientists that are very important. So guys, these are the landmarks in the history. We already studied it, and so we are jumping few pages. I'm sorry, it's the previous one. Here, don't do this to me. So the conflict over the spontaneous generation. Now they are telling us the story, like how was the conflict happening on the spontaneous generation before going into the detail about the golden age of the microbiology. Now, okay, I didn't have to explain that to you guys. Wow. From the earliest time, people had believed in the spontaneous generation that living organisms could develop from non-living matter. Even Aristotle, wow, he is ancient, 384, 322 BC, thought some of the simpler invertebrates could arise by the spontaneous generation. Okay, so even he thought, because It was the invertebrates, means they were on the micro levels, unlike the other invertebrates. So I can understand if he thinks that, yeah, their reproduction comes just out of nowhere, because they were, maybe their eggs were just so small. This view finally was challenged by the Italian physician Francesco Redi. Now here comes the Francesco. That Francesco that developed the microscope was a different Francesco, and this Francesco is a different one, and it is in 1626 to 1697. Wait, was he really different? Yeah, he was different. I don't remember reading the name already. Who carried out a series of experiments on the decaying meat and its ability to produce maggots spontaneously. So it wasn't the mosquitoes. Okay, sorry, I was calling them wrong names. Maggots. Reddy placed the meat in 3 containers. How did he challenge this spontaneous generation? This is the process. First, he took the meat and put in the 3 containers. One was uncovered and the second was covered. Wait. What's happening? Am I getting angry? Am I? Let's smaller. Let's make it small. No. Let's enlarge it. How are you guys going to be able to see this? Wait. Come back. What the heck is happening? Don't make me angry. Okay. Whatever, I'm just telling you, okay? You don't have to look at that. One was uncovered and the second was covered with a paper. And the third was covered with the fine gauze that would exclude the flies. Flies laid their eggs on the uncovered meat and the maggots developed. The other two pieces of meat produced the maggots spontaneously. Okay, so they are saying the one that was the uncovered one, right? So he said that, yeah, the maggots came and laid eggs, and yeah, the maggots came out, right? But what about the other two? The one was covered, means there was no space for the maggots to come and lay eggs, right? But still they produced the maggots. What about the fine gauze? He was still, means like acting like a filter, but still the maggots were produced. However, the flies were attracted to the gauze covered container and laid their eggs on the gauze. These eggs produced maggots. The generation wait. Me did not. What was I doing? What am I doing? What am I saying? Oh, it was the fault of these pictures going up and down. It wasn't my fault for getting it wrong, even though I know what the spontaneous generation was and how was it rejected. Okay guys, I will cover it. There were 3 containers. One was uncovered, one was covered with the fine gauze, and one was covered with the paper to seal it. So the maggots come from the uncovered one, which is okay because they laid eggs and they developed. By the way, the second, on the gauze, the maggots came because they were trying to go to the meat, but the gauze was in the middle of it. So, they just laid their eggs there. And the third one was sealed, and so the third one didn't have any maggots because no eggs were laid on the meat. And also, I think they already, sterilized the meat, because general meat has a lot of microorganisms as well. These are maggots, not the microorganisms. So the eggs that were on the gauze also developed into the maggots. So the third generation of maggots by decaying meat resulted from the presence of fly eggs, and meat did not spontaneously generate maggots as previously believed. So he just said that, yeah, if the life could come out spontaneously, why did it not came out of the third container? like if your theory is correct. So that's how he refuted the theory. Similar experiments by others helped discredit the theory for larger organisms, right? Now, Leeuwenhoek's discovery of microorganisms renewed the controversy. Like, now there comes the Leeuwenhoek. We remember this guy, right? The one that was working in the accessory shop and the one that was developing microscope in his free time. His discovery again started the controversy, which was almost dying because of the scientists that previously refuted the spontaneous generation by their experiment. Let's see what he did to arise the controversy of the people again. Some proposed that the microorganisms arose by the spontaneous generation, even though the larger organisms did not. Okay, some believed the spontaneous generation theory, some didn't. They pointed out that the boiled extracts of hay or meat would give rise to microorganisms after sitting for a while. Okay, so they were clear about the larger organisms, right? Like larger organisms cannot come out on its own. The larger organisms can't have the spontaneous generation, but they were still not believing that the smaller microorganisms can't have the spontaneous generation as well. They were thinking, yeah, they maybe microorganisms can do the spontaneous generation as well. So, how did they point out that they can do that? That the boiled extract of hay or meat would give rise to microorganisms after sitting for a while. Okay. Normally, when we heat up the meat, usually the microorganisms are killed. Not usually, yes, they are killed, because even after you put it in a pressure cooker or steam, not steam, when you cook it, after with the high heat, the microorganisms get killed. That's a true fact that we still believe till this day. But they were saying that when they boil the hay or the meat, it still gave rise to the microorganisms after sitting for a while. Why was that? Why was that? And it's because Leeuwenhoek misunderstood the temperature requirement for killing the microorganisms. He didn't know that the temperature requirement can change the killing of microorganisms, like the ability. Usually there are levels of heat required for different types of food, and the meat has been, needing the highest amount of temperatures to kill that bacteria. So it's either that he misunderstood the temperature requirement to kill the microorganism. It's that, or it's because of the spores. There are bacteria, like I think there were two. I almost completely forgot their name. Sorry about that, but still, there are some bacteria that has spores, and when they lay the spores in the food, they can't be killed by just simple heating method or cooking method or boiling method. So it was either the spores or it was either the misunderstanding in the temperature requirements. So they were almost, believing that the spontaneous generation was incorrect until Leeuwenhoek discovered the microorganisms in the boiled extracts of hay or meat. So what happened next? In 1748, the English priest John Needham, 1713-1781. Okay, this is his lifespan. Wow, he didn't live long, right? 13, 23, 33, 43, 53, 63, 73, 83, 60, almost 70. Wow, he lived a long life. I was wrong. Reported the result of his experiments on the spontaneous generation. Needham boiled mutton broth. Okay. So the Leeuwenhoek did his job and raised the controversy again, but John Needham again did the same experiment, and then he tightly stoppered the flask. Stoppered means he sealed it with a cork or maybe something. So he wasn't like, Leeuwenhoek just boiling and leaving it open. He first sealed the flask and then he boiled it. Eventually, many of the flasks became cloudy and contained microorganisms. So, even though he stoppered the flask tightly. Still many of the flasks, not all the, and they already contained microorganisms. He thought, sorry, I forgot that you guys couldn't see the lines. I always forgot that I have to bring it up. Eventually, many of the flasks became cloudy and contained microorganisms. He thought organic matter contained a vital force that can confer the properties of life on non-living matter. Whatever, what the heck? He brought out the even wild concept. He said that maybe organic matter had a vital force that could inflict the non-living things and give them life or something. Oh my gosh, oh my gosh, oh my gosh. That was so wrong. A few years later, the Italian priest and naturalist Lazzaro Spallanzani. Now there comes a priest. How are the microbiologists so diverse? One was working in the accessory shop, or the men's wearing shop, and now the other one is a priest and a naturalist. Good thing. Spallanzani, I know this name. And you also remember it because he is an important guy in the microbiology. What did he do? Improved on Needham's experiment designed by the first sealing the glass flask and that contained water and seeds. If the sealed flask were placed in the boiling water for 3 to 4 of an hour, means how do you call it? Almost one third, no. Okay, we get it, 3 by 4, right? We get it. Like if you divide it into 4 parts, there's 3 parts of oh gosh, means he 30 minutes and then with the addition of 15 minutes. 15, 60, so wait. 30, 40, 45. He could have just wrote 45 minutes. What do you mean by 3/4 of an hour? Gosh. No growth took place as long as the flask remained sealed. As long as it was sealed and as long as it was boiled for 3 by 4, I mean the 45 minutes, everything was okay. He proposed that the air carried germs to the culture medium, but also commented that external air might be required for the growth of animals already in the medium. So he said that maybe the air had the germs, so that's why, because he sealed the flasks, no microorganisms grew. But then again he said maybe the microorganisms didn't grow because they needed air, and he completely cut off the air from them. Means they needed oxygen and they couldn't grow without oxygen. So he said 2 lines, but completely opposite of one another. First he said that maybe the air had the microorganisms and he completely sealed, so maybe the microorganisms couldn't enter. The second, he said that maybe air was needed for the microorganisms to grow, just like animals, right? So maybe because that's why he didn't grow. So he wasn't completely sure what was the thing that was stopping the growth of the microorganisms, which we know these days that it was the heating process. Because microorganisms are present everywhere, whether you seal it or you don't seal it. Even if you seal it, it's already in them. So the supporters of spontaneous generation maintain that heating the air in the seal flask destroyed its ability to support life. Okay, they are still stuck. They thought that air had the microorganisms, and now that the air that was sealed in the flask, when we boiled it or when we heat it, that lost its ability to support life. So they are blaming the heating of the air, not the heating of the microorganisms itself or the life itself. Several investigators attempt to counter such arguments. Theodore Schwann allowed the air to enter a flask containing sterile nutrient solution after the air had passed through a red hot tube. Okay, now he was showing that I am heating the air, right? I am heating the air. If I heat the air, but I am leaving it open, and the nutrients are clean as well, he boiled it even before that, and he hotted the tube as well, and the air passed through it. The flask remained sterile, means nothing happened. Subsequently, George Friedrichs Schroeder and Theodor von Disch allowed the air to enter a flask of heat sterilized medium after it had passed through the sterile cotton wool. No growth occurred in the medium even though the air had not been heated. Okay, so air wasn't heated. What am I saying? So they sterilized the nutrient, but they sterilized the material as well, which was the red hot tube in the case of the Schwann, and it was the cotton wool in the case of Schroeder and Dish. So they said that they didn't. What am I saying again? The Schwann had heated the air, right? But still no growth occurred. So Schroeder and Dish said maybe if we don't heat up the air, it will cause growth of microorganisms. So that's why they put a cotton wool in that. So cotton wool is a bad conductor of heat, so when the air passes through it, it can't be heated. So even if the non-heated air enters into the medium, nothing happens, no growth occurs. Despite these experiments, the French naturalist Félix Pochet claims, is it Pochet or Pouchet? Pochet, claimed in 1859 to have carried out the experiments conclusively proving that the microbial growth could occur. Wait, guys. This thing here. I think we should. I'm talking about the Google window. I think we should make it smaller because we don't need it, and I'm covering from you guys the statements or the sentences. Have carried out the experiments conclusively proving that the microbial growth could occur without air contamination. Now he proved it, the Pochet, in 1859. So they were playing around centuries after centuries just after the spontaneous generation. Are they kidding me? This is already 1859. And now the Felix Post, oh guys, it's Felix, claimed in 1859 to have carried out experiments conclusively proving that the microbial growth could occur without air contamination. Now he is saying that you guys thought that air had microbes in it, and it couldn't support the life because it was killed, and even if we don't heat it, the microbes didn't grow. But this guy said no, air isn't the main thing. Air isn't the main thing. What are you guys talking about? Like air, heating the air or things? So I think he conducted the experiment without involving the air, and like I told you, microorganisms are present in the food whether you contaminate it with air or not. It has always been contaminated with microorganisms. Even our own body, which you see like it's clean and everything, our every cell, every pore has millions of bacteria or microorganisms on it which we cannot see. So of course food is no exception. So this claim provoked Louis Pasteur. Oh gosh, Louis Pasteur said, what did you say? How do you ruin the work and belief of the past people? Let me do something for you. Let me settle this once and for all. By the way, Louis Pasteur is the father of modern microbiology, and he did a lot of things. He did a lot more than this Leeuwenhoek, which oh my gosh, what can I say? Should I blame him? Yeah. First he just saw the microorganisms and stirred the world, and then he did what? He stirred the controversy again. But he is still called the father of microbiology since he discovered the microorganisms for the first time. And he did a big thing, and because he published it. Unlike a lot of us that are too lazy to publish things, even if we do. So provoked Louis Pasteur. Now here comes the Pasteur. To settle the matter once and for all, Pasteur first filtered the air through the carton and found out that the objects resembling plant spore has been trapped. Okay, he filtered the air through the carton. Guys, I didn't know air could be filtered through carton. No one told me that carton is that much of a good filter paper can act as a good filter. So he said that the plant spores were trapped and the air was clean. If a piece of the cotton was placed in a sterile medium after the air had been filtered through it, microbial growth occurred. Okay, so they saying that if the same piece, right, of cotton which had the air passed through it, filtered through it, they said the microbial growth could occur. Like, yeah, air had something in the air was there to cause the growth. Now next he placed the nutrient solution in the flask, heated their necks in the flame, and drew them out in a variety of curves while keeping the end of the necks open to the atmosphere. Pasteur then boiled the solution for a few minutes. Okay, right, the previous experiment had two things, right? Two things, and the two things were nutrients and air, and people were blaming everything on the air without looking at the nutrients. And this guy said, no, let me clear all of this, let me take everything one by one. And he cleared that the air had the something that were filtered and still caused the growth. Now he went towards the nutrient, and he put them into flasks, multiple types of flasks, and then he heated them for a few minutes, and then he let them cool. No growth took place even though the contents of the flask were exposed to the air. Yeah, he didn't sealed it, and he already proved that the air had something that can grow in the nutrients, right? But when he kept heating that, even though the air was there, no growth was taking place. So Pasteur pointed out that the no growth occurred because the dust and germs has been trapped on the walls of the curved neck. So now he is saying that even if I heat the nutrients, heat the nutrients, but the curvature stopped the microorganisms because the microorganisms stuck to that curved necks. So if the neck was broken, growth commenced immediately. Like I told you, he allowed them to cool. If he kept boiling it for days and days, no microorganisms would ever grow, even if he kept boiling it even for a year, right? But he let them cool, right? Cooling was the problem. When you stop heating and the new microorganisms go into the same nutrient is safe. So of course they are going to grow because who is heating them? So that's why when the necks were okay and working, the microorganisms couldn't go into the sterile medium, and yeah, no growth could take place. But when they were broken and air was directly into the contact with the nutrients, the growth occurred. So Pasteur had not only resolved the controversy by the 1861, but also had shown how to keep solutions sterile. So how did he resolve the controversy? Like, air had the microorganisms, air had something in it that could grow, and people already believed that. And there was also something in the nutrients that could grow if we don't heat it. Like, both had something. It wasn't not just the air and not just the nutrients. And boiling was the problem. Like, it could kill the microorganisms. And by the way, did you guys notice his name is Pasteur? And have you ever heard of pasteurization? I will give you example you guys know in daily life. I think, yeah, milk pasteurization, right? The milk is, milk goes through high temperatures, extreme high temperatures and pressure. So high temperature for killing the microorganisms. Okay, now what was he doing with this? What is this? Focus screw, specimen holder, lens, lens, specimen. Where will he see this? Handle. What is this? What the heck? A, B, and what is this? This is bacteria. This is also bacteria, and this is streptococcus bacteria. Strepto or staphylo? Coccus. These are cocci, chained cocci. Sorry if I'm saying it wrong. I don't know if it was strepto or staphylo. Wait, let me search. I don't want to be wrong about this. Which one is coccus? Oh, okay. Staphylo is chain and strepto is clusters. Which one is chain? Strepto or staphylo? Right. Right. What? I was wrong? See, that's why I'm saying I'm mixing them up. Strepto is the chain one and staphylo is the cluster one. Let's see the diagrams. Even though I know it, you guys can see it. Like these are the chains and these are the clusters. These are. So let's go back. And I don't understand what he was drawing with this one. Is this the helminth? Or is this, no, it can't be Vibrio, right? Vibrio is just a comma or, and what are these? I think something will be down. And I think this is the microscope. I didn't know the microscope would look like this. The lens is just so small. And what is this thing? This. What is this? This arrow. Oh my god. Okay, so we are done with this for now. Okay, they have, written it here. I'll show you guys as well. An oil painting of Leeuwenhoek. We know that. And a brass replica of Leeuwenhoek microscope. So this is Leeuwenhoek microscope. The microscope Leeuwenhoek. So this is what Leeuwenhoek made. It's so different from the current microscopes. Leeuwenhoek's drawing of bacteria from human to human, from human mouse. Drawing of bacteria. Guys, I see this is bacteria, like Bacillus bacteria, and this is the Streptococcus. But what is this and this? I think this is the Vibrio one, right? It's like curved. This must be Vibrio. But which one is this? What are these? And this says that he isolated them from the mouth. Of course, our mouth has a lot of them. But what is Leeuwenhoek's photo doing there? We were talking about the Pasteur, the great Pasteur, who finished the controversy. Here he is. Here. Pasteur working in his lab. Guys, look at him. Guys. His forehead says that he is intelligent. His eye says that he is intelligent. Wow. Here are the flasks which he used. Wait. Let me show you. So this one is curved like this completely. This one is like this. These one are carved like this. And all of them. Pasteur's swan neck flasks used in this experiment on the spontaneous generation of microorganisms. Source: Annals Science Natural, 1st season, volume. Is this a book? So anyway, these were the flasks. I didn't know they had this much variety in them. And we should look for our live as well. Oh my goodness, why did I? Is it working? I just want to know. Should we move further? How much is remaining? I wanted to get done with these scientists, but I think we will have to go through it a lot because we are still in 1820. What is this? The English physicist John Tyndall in 1820. This guy was doing all of this in 1867 or something, but deal with a final blow to spontaneous generation in 1877 by demonstrating the dust did indeed carry germs, and that if dust was absent, both remained sterile even if directly exposed to the air. Like if you clean the air instead and then give that air to the sterile, the no growth of microorganisms would be done. During the course of his study, Tyndall provided evidence for the existence of exceptionally heat-resistant form of bacteria. Right. These were the bacteria I was talking about. Working independently, the German botanist Ferdinand Kohn discovered the existence of heat-resistant bacterial spores, endospores. Like I told you, these are the spores that cause the bacteria to resist the high temperature. That's why they are heat-resistant bacteria. And are we done with this? And they said that they will be explaining the bacterial endospore in section 3.11, means it will be in chapter 3. We are not going to go for the golden age. Let's leave it at that. How about here? Time to say goodbye. We are done with the live, and I think we are okay with reading this much. And I think I talked too much about explanation. Like, no one is stupid enough to need explanation in the discoveries. So if you guys don't need it, you guys can comment on it. And we are going to study all of these. We are going to go through Coach. This chapter is all about the scientists. I underestimated it. Right, I underestimated it completely. Right, look at these, the future of microbiology. Okay, it's such a long chapter and it has scientists, and I will be summarizing all of this means so we can remember what we studied. All right, this is